All right, everyone. So hopefully you're in the right place if you're here for the information session for research experience and learning. We call this group Real STEM. It flows off the mouth pretty easily. So rather than saying research experience and learning STEM coalition, it's Real STEM. So who's talking? Me. And my name is Anuya Banerjee. A little bit about me. I am the club president for this group. Just happened to be right place, right time, right motivation. And so, fun fact, third year student currently at ARC, full-time student. I've actually been taking classes here for about 7 or 8 years now. So that included my time during high school when I was taking classes. So this is really my second home. I feel welcomed here. I hope you guys do too on this campus. And yeah, so I'll mostly be talking to you today. We of course have our, so if I'm president, he's vice president. So please introduce.
My name's Logan. I have not been here as long. I am also a third year student, but I've only been part of this club for about a year now. So that's kind of my involvement. I got in last year. Same thing, heard it from my bio classes, was interested, wanted to get part of it, right passion, right time. So yeah, we've had fun with it.
Yeah. And then we have our advisors who let us do everything we can. So they're really the powerhouse of the cell here. So we have Dr. Talene.
Hi, I'm Adam Talene. I've actually been teaching here at American River College since 2007 in various capacities. Large chunk of the time since then, though, I was teaching at Sacramento State as a lecturer, teaching genetics, molecular biology lab courses, including a bunch of classes for their stem cell master's program. And I was doing that and just moonlighting here at ARC, teaching a class or two on the side until 2018, when I took over our biotechnology program. So coordinate the biotech program and teach most of the courses, right? And you're here in the biotech lab. And you'll probably hear lots more about me later in, as we go on. So I'll cut it short here.
Yeah. And then we do have one more advisor. She's a teacher in the chemistry department. Dr. Becker over there, but she's not here, but her name is written on the board. And she might be joining us later. We'll see. Otherwise, we'll see her hopefully at the next meeting or so. And yeah, so she helps us with the chemistry portion and guiding us in that aspect of our water research. So agenda here, it's written on the board just so that you know where we're at as we go through. We'll talk about the history, our goals. At the end, we know some of you, we wanted to make this as open as possible because a lot of diverse minds really helps this project actually. So at the end, if you don't have any micropipette experience, we do want to pull those out. We'll take a little break in between, have you guys introduce yourselves, and then we'll get going from there. So, oh, and real quick, this is a lab class. We do want you guys to stay hydrated and everything, drink your morning coffee. If you do need a drink, please step outside real quick or do whatever you need to do. Just please don't do it in the classroom because there's a lot of lab stuff going on here and we do not want to be drinking or eating in the same room as people who are doing some certain experiments in here. So.
Oh yeah, and we have plenty of biohazards and hazardous chemicals in this room, right out on the benches where you're all sitting with your stuff. We try to clean it up, but just as a precaution, remember this room is a laboratory.
Yeah. All right, so clicker. We'll get into it then. So starting off with the goals. So some of these are from the student perspective, some of these are from the professor perspective. So for Dr. Talene, his original motivation was to really create opportunities for research for community college students. Because historically, as you guys may know, once you transfer to like the UCs per se or four-year universities, there's plenty of research opportunities over there. But at the community college level, that's historically been an underrepresented area. So in addition, he also, as he mentioned, he teaches the biotech courses, and one of those is bioinformatics, which can actually benefit from some of the data sets that we create during this project, so they can actually be used in the classroom setting for the classes that he actually teaches. And then this is really a goal on the student side, collecting data about our namesake waterway. So of course, American River College is named after the American River water, and what we as students want to do is contribute to an ongoing data collection and contribute to this framework that's going on so that not just you can participate on research and, finding out information about prokaryotic diversity in the water, but also students who come after you. So we're gonna see things hopefully over time as this project continues to develop. And, if you think about it, the landscapes are constantly changing. our waters are not the same as they were 10 years ago because of things like climate change, human development. And so we can actually help to see those changes over time with our research. And then this is kind of like an older goal because it's already been done, but create opportunities for collaboration. So now we have a chemical portion to this research where we analyze the chemicals in the American River water. And that's with the help of Dr. Becker's expertise, of course. So it's no longer just biology and biotech. We've expanded that, but there's always room for more collaborations. So for example, geography students, if you guys know any, they can potentially help us with determining sites and mapping out some of the places that we've been to, seeing the changes in landscape between them. So that's actually a great opportunity for all sorts of people to get to join in.
And if you got ideas, we'd love to hear them.
Yeah, exactly. These are just some pictures of our lab work that we've done. But so hopefully you guys saw on the flyer the terms metagenomics and microfluidics being used. And hopefully you guys looked it up. But if you weren't, maybe we all want to be on the same page here. I'm just going to explain it real quick. So metagenomics is the study of genetic material recovered directly from environmental samples. So that means, you know, that could be a soil sample, for example, but it doesn't have to. In our case, it's actually water. And we're not just looking at one organism in that sample, it's actually all of them that are in there. So to figure out bacterial species diversity in the sample, we actually look at the ribosome. And maybe without anybody seeing this, can anybody tell me what the ribosome does? Yes.
Translates protein.
Yes, exactly. So this is the molecular machine that is spitting out proteins in the cell. So, and again, to be on the same page, so 16S ribosomal RNA, that's the functional part, the actual ribosome that translates and makes proteins. And that's actually encoded by a section in the genome. So the gene that encodes that would be 16S rDNA. So you might hear those terms being thrown around. So on the same page now. And here is a linear piece that represents the 16S ribosomal DNA. And you can see these green regions. So those are regions of the DNA that are highly conserved. So across prokaryotic species, that region of DNA in their 16S ribosomal DNA will be pretty much the same. Any prokaryotic species that you're looking at, bacterial species. Now interspaced in between those are these V variable regions, they're highlighted in gray. And it goes from V1 to V9. So those are basically regions that are specific to a particular organism. So just to give an example here, if you have E. coli, which is in the water, fortunately or unfortunately, if we take the V4 region in E. coli and we sequence it, in E. coli, that will actually be a pretty similar V4 region across all of them. So that's what I mean is we can use sequences of the variable regions to basically barcode certain organisms. And then if we take that 16S ribosomal DNA and we amplify it, so that means make several copies of it through a technique called PCR. If you've taken biology, you probably know about this. If not, we're going to go through a lot of these techniques over the semester and you'll learn it as we go. But basically, we'll make several copies, we'll help barcode them a little more, and we'll sequence them. And then that will associate with a particular organism when we go through the databases and see, hey, what do we have in this sample? So that's kind of the basics of metagenomics. I'll hand it over to Logan to talk briefly about what microfluidics is.
Hi. So I'm going to briefly go over this because actually in our research over the past year, we've put a lot of our efforts into the metagenomics, but microfluidics is definitely something that we continually want to do. So what is microfluidics? It's basically just analyzing water using small channels and usually chemical reagents of some kind. Especially in here, we're using colorimetric reagents. These are things that will react with certain things in the water to show an actual change. If you've ever taken your Gen Chem 2, this is like your Beer's Law stuff. You put something in there, it's mixing with the chemicals in it, reacting and causing some kind of color change. So the things that we are looking for specifically with our research is what could be in the water and what are things that are affecting the waterways through like climate change or human development. So this includes things like iron, copper, nitrates, urea, phosphates, other things that we can find them. If you have ideas that you're like, this would be a really cool chemical to find and really important, we would love to hear it. So again, this is just kind of briefly what we've seen, briefly what we're doing with it. And I will hand it over to Talene now to go over history.
Okay, so I want to just talk a little bit about the history of how we got here. So we've actually been working on the metagenomics portion of the project for quite a number of years now. So we started, actually the original idea for the project came up in 2018. So this is all pre-pandemic, just by accident. So I took a group of biotech students to the Halloween symposium at UC Davis at their Genome Center. And one of my students, pictured here, Laura Leskinen, actually won a raffle. And what the raffle was for, it wasn't like a monetary prize, it was free core services from the Genome Center. And so we started saying, well, what could we do with this? What kind of project could we do in the lab where we could utilize some of the sequencing resources? And so that got us thinking about ideas for this. And 16S metagenomics was pretty hot at that time. So we thought, oh, that might be a good thing that we could actually do here at the community college. And that would, we have most of the equipment. I mean, we'd have to send the DNA out for sequencing, but we could isolate and prepare all the samples for that. So now we didn't end up using her raffle money or her raffle, core services for that. She ended up using it for training, but that kickstarted the entire idea of this project. So then fast forward to the next summer, I put together a small proposal to get a little bit of funding to kind of pursue this idea because it seemed like it was a good idea to sample microbes from the water, right, and get students involved in research. So that was funded. I got a modest amount of money for that. And then that led me into fall 2019. I was having my office hours over what used to be the Mesa Center or the Science Center. And I ran into some of the students there. And I started talking to Natasha Skidmore and the Women in STEM Club, and that's when Dr. Becker got involved. And so we put together an innovation grant proposal to get a little bit of funding, to kind of do this for the DNA sequencing, right? So we could get more involvement, right? And that brought in one of the things I wanted to do, which is make it more interdisciplinary, right? We got Dr. Becker involved to add a chemistry portion, right? You can see Natasha right here. She's now down at in Southern California. So after a little bit of development in spring 2020, we were finally ready to get started in the lab. So here you can see we were over in one of the chemistry labs. We were playing around with microfluidic devices, doing things like melting crayons on filter paper to see if we could, make hydrophobic barriers and get the chemicals to go through the channels like we wanted. One of the things we also did, you can see students at the time melting crayons. We've since abandoned that. We found some better options, but you don't know until you try it, right? And that's Dr. Becker pictured in the back there in the green sweater. But then we got shut down by COVID-19. And actually over here, you can see some of our students at the time. And this might look like a tire pump. That's actually what it is. One of the things we're going to have to do is get the microbes out of the water so that we can isolate DNA and work with it. So the easiest way to do that, there's the microbes are pretty dilute in the water, right? If you take a liter of water, most of that's water, not the actual microbes. So you have to get them out. So the easiest way is filtration using a vacuum filter. But we wanted to go collect water, places where we couldn't necessarily run an electric vacuum pump. So we thought, well, wait a minute, could we get something that's handheld that we could like hike out to sites on the river? Because some of my motivation for this project was being able to go collect water like up in the mountains in remote areas, right, and analyze some of the microbes present there. So we actually got a car tire pump and we modified it and flipped the check valve around and turned the tire pump into a vacuum pump. And that's what we're currently using, right? This actual one here that we modified. However, right as we got that working and we're starting to think about actually going out and collecting samples, right? Probably everybody remembers spring 2020, not the greatest semester. So that shut us down. And then we spent several years where we basically couldn't do anything in the lab. So we continued to still meet, try to develop the project conceptually, but we were mostly reduced to Zoom meetings and bringing in guest speakers and, like journal club style, like reading relevant papers and things like that. But that was enough to keep us going until we could finally start getting back in the lab. And I think that's my last slide, right? So I'll pass it back to Anuya to talk about post-COVID.
Yeah, and we'll show you some of the materials that we have now. We still use the tire pump. It's gone through a lot, I'm not going to lie, but it still makes do. So fall 2022, that's kind of when I joined in. And I'm not sure if I said this, but I was kind of like the first which I like to say generation of recruits who got their hands working on some of this lab work stuff in terms of really pushing metagenomics further. And then Logan, second generation, you guys would be third generation. So we all are the people who get to work on the water. And in terms of what we did this semester, that was actually a lot of training. Of course, that was the first time we could come back into lab and do this type of work. And so it was a whole semester of training. We're not actually going to be doing as much as we did that semester, just because a lot of these we have our project pretty well streamlined at this point, and we can just move forward with applying these techniques to our project. So you'll learn them as we go. And then spring 2023, we made a lot of progress this semester. And some of the things we had to do was order our materials. We actually hadn't had that except for the tire pump. But some of the things we had to do was order a DNA isolation kit. And we had to do some literature research about this actually, because as you can see, we chose the Power Soil Pro kit here, and we're using water samples. So why are we doing that? Through research, we discovered that for isolating DNA from environmental samples, this is actually the best kit to use. It is a little expensive, but it works the best because when we think about soil versus water, soil has a lot more organic material, nutrients, just gunk. And actually, water has a lot of gunk to it. This is like a flask from the creek water right around here. And that's pretty dirty over there. You can see it's settled quite a bit. This was definitely not the greatest color, most appealing color beforehand before it settled. So for that reason, the soil kit is actually highly effective with water samples and getting all that stuff out and isolating our DNA and purifying it. So yeah, me and another group member did a lot of research. Did a lot of research on that, sorry. And we found some analytical reagents for our microfluidics. We also developed a lab notebook. So this is something that was kind of novel that semester. It's really important that past people who are involved with this club can show what they've been doing, some of the progress they've made, so that now you guys can look back at whatever we did on a specific meeting day in the past in 2023 and know exactly what we did. And so we'll keep that up this semester so that future generations can continue seeing what we've done. And then we also did a proof of concept poster. So again, we were ordering our materials. We actually haven't had them yet. And we did a proof of concept poster. So no research presentation, but kind of our idea for what it would be. I think the beginning slide has a bigger picture of it. So we did a metagenomics section of the poster, kind of what we thought. This is actually like some stock images of the glassware that we have now. Like at that point, we really didn't have anything. And then a microfluidic section. And that poster is elsewhere because we've done more since then. But yeah, so we presented on that. That was really fun experience for me as well. And then we developed our microfluidics. So again, before 2020, the past group was actually working on this, but since I wasn't a part of that and we didn't really have anybody to, except for the professors, to vouch for what type of logistics had been done before, we were kind of starting from scratch basically. And so just to get you guys up to speed on where the microfluidics is at now. So we started off with our device being on just a qualitative cellulose filter paper. And what we wanted to do was create a hydrophobic barrier so that we could place our liquids onto it and have it be evenly spaced. So when you put an analytical reagent plus an analyte, you want them to be contained, because if it actually spreads out, the color of that solution will actually change based off of how spread out it is. So in a smaller well, it's going to be more concentrated and therefore more colorful, or more intense of a color. And so with that hydrophobic barrier, we tested a lot of things. You saw the picture of them melting crayons. We tried markers. We tried, there are these colored pencils that are kind of a little bit more lipid based. So we tried those, but we actually landed on a whiteout pen. And, I didn't have the best success rate. I want to say it was about 70 or 80% of the time it worked. So not always, but we were able to compromise and say, okay, if you use a small amount of volume of your liquids, it would actually work most of the time. And we actually had this is an example of our filter paper with the wells and the whiteout It's kind of hard to see, but we actually had one of our previous members 3D print like a way that we could evenly put wells and trace out circular barriers onto the filter paper. So that was really great. I still have that with me, not here, but it is with us. And then the analytical reagent that we're using was ferrozine. So this creates a chemical complex. If you take General Chemistry 2 or have taken it, you'll kind of learn more about these complexes that are made. And this detects iron. So here's actually a picture of different concentrations of iron in these standards. So 0.1 all the way to 3 parts per million of iron. And when you have ferrozine in the sample, it creates this color complex, kind of like a pinkish color, which changes the intensity of the color based on the concentration of iron. So we can use those standards in liquid. We can find the concentration of an unknown sample via spectrophotometry. So again, if you've taken biology 400, chem 400, chem 401, you've probably used those. But we're trying to translate this into microfluidics. So on a paper-based device, we actually have to do this based off of color intensity on the paper itself. So how do we do that? How do we associate color intensity with concentrations when it's basically image-based? And the answer to that would be through software. So you actually have to take a picture of those samples and then use software to figure out what are the concentrations. So this is actually a proof of concept here. So obviously this is a digital like colorized thing, but those colors were mathematically determined to have like a relationship. So this unknown here is actually 2.0 parts per million of theoretically iron. And we were able to show that this concept worked to use ImageJ as a software to figure out unknown concentrations based off of color intensity. But the problem is, even with this perfect proof of concept that we can use our phones and use, software to figure out unknown concentrations, the problem with that was that based off of several factors, it's going to change each run that you do with it ever so slightly. So for example, we printed out that perfect color palette, theoretically perfect, and we were taking pictures of it and then uploading it into ImageJ. But depending on the lighting, depending on the phone camera used, these were all creating like about a 14% error with each find of the unknown concentration. So like 1.9 ppm, 2.1 ppm, when actually it was 2.0 parts per million. So little things like that. We've tried really hard to like, we literally created this box here and we shined a ton of light into it, but even so shadows were being created. So this is where like photography students, people with an interest in that, or just creative thoughts, we want to pursue this. And that's kind of where we left off with the fact that we're having problems with imaging and we're having problems with understanding how to standardize this process so that all samples are created equal. And so that's where we left off. Again, we want to pick that up. But summer 2023, that's when we actually got our materials and we got onto sites. There's a lot of fun backstories. I probably shouldn't tell it right now, but so yeah, just lots of lab work, lots of fun. And then this is of course when we started another round of recruitment. So Logan is actually the best person to talk about all this stuff that we've done since fall 2023.
So yeah, fall 2023, this was when I joined the group, so our second generations. This is when, they had done a lot of the work to kind of get us to where we needed to be. And so this semester, we really wanted to emphasize just building a framework. We wanted to be able to collect samples, do it fast, get a lot of data, be able to present that data eventually. So we started doing testing different versions of our protocol. This meant like, as we started going through the process, we started realizing some things aren't going to be as smooth as we once thought they were, so we need to smooth them out. So this is what that meant. One of these examples was like, we kept getting weird spikes when we would go do DNA concentrations, but we were like, this isn't DNA, what is in the sample? So we just moved our, I guess our techniques around that, and we developed them a lot this semester. And then the last thing that we ended up doing was our Sanger sequencing, our poster presentation. The poster's actually up on that wall over there. And it was a lot of fun to do. We got a lot of samples. We got to see a lot of cool microbes. one of the things that we saw that was actually really interesting is that when we put these back into our BLAST database, which is an NIH database, we came up with a lot of microbes that we're seen like all over the world, but we're in our waters. And that's cool because it's like these are regional species that we're seeing here. So a lot of interesting stuff. So just a bit of like our workflow for what we kind of figured out. The first thing that we start with is collecting water samples. Of course, we need something to run our samples on or run things on to get data. This begins with autoclaving all our gear. We want to make sure that we aren't getting any contaminants in there. We want to make sure that what we're getting from the water is actually from the water. It's not from our lab or something else. This then went to our filtration with the vacuum pump. You see that here, actually. That's our tire pump that we've modified to suck instead of push air through. And then one of the things that we figured out was preserving filters. What we thought at first is that we could, do these filters, run water through them, collect microbes, and then store them. That isn't the case. If we stored it in ethanol, you see it becomes this disgusting jelly and is impossible to do anything with. Instead, we figured out it's just better to store them there, get them back to the lab, and then do the processing immediately. Oh, and really quick, one of the things that we're really trying to look out for, since this is a full environmental study, we want to make sure that we're having good observations. We want to know like what the water quality is, where we're collecting the water from, local wildlife or human activity. One of the things that we found actually this semester was like we did water samples near where they were doing construction near Sac State, and we did find microbes that are common in high iron concentrations, high water concentrations of iron. And that's really interesting because they're doing construction right there. So what does that mean? Things like that. And this was a small one. Why does the water bubble in the flask? When we're pulling a giant vacuum, it starts bubbling up. Just things to think about. So next we get back to lab, right? We have our samples on this filter paper and we need to process that. This is basically our power soil DNA extraction kit, which is the thing that you showed earlier. It's really helpful. It kind of runs us down everything we need to do and really gets us through all that. And then after we have that, we basically have a portion of DNA and we usually run a gel of that to make sure we actually have DNA. It's not something weird. Next, we go into basically our portion of what we call our library construction. This is where we need to amplify our DNA, basically purifying our samples so that we know what we have and what we're going to send off or what we're going to sequence later. DNA sequencing. This year we sent off our samples to get Sanger sequencing. This year we may be getting a sequencing thing here. Hopefully, it would be really fun to have that. But yeah, and then in the future we're going to do next gen sequencing. I'm not going to go into that much now, but it is something that we were thinking about and would be a slightly different process. Next we do data processing. So this is, we get back our DNA samples. Basically, they have been sequenced. We have the pure code. What is this? And this is where we need to kind of do some extra things. And that would be like cleaning, trimming raw data. Sometimes you get messy things. If you ever look at this, you basically get back this massive data that is just a bunch of ACGTs and it's all clumped up together. So we need to do a few things about that data. But then we are able to actually analyze that data. It's all good. And then we can actually look, what bacterial species are we finding? Where are we finding them? Why are we finding them there? Things like that. And then, yeah, different species diversity, and then we combine with other data. So water chemistry and vertebrate surveys, which is things that we haven't really started. We've done a bit of our water chemistry. But yeah, eventually we do want to do full environmental workups. We want to understand what's happening in these waterways. And yeah.
Okay, yeah. So just to kind of recap what that means for you, really our workflow that we'll be starting out kind of mid-semester is, it's going to be pretty repetitive, but we're going to go to different sites. We're going to process it in the lab. We'll check our DNA quality via gels and spectrophotometry to figure out the concentrations of DNA. And, when springtime comes, that's when we'll start focusing on how do we sequence this DNA that we have now? How do we, get some results back? And pretty much every spring so far, I don't know if they plan to do it again next spring, but another, we get the opportunity to present a poster at a event called the Serious Conference. So we basically get a little bit of their grant and recognition to be in their grant funding. And so that's at Sac State, actually. It's this grant that helps promote research opportunities in the classroom. So we don't fully fall under that because this is not a class, obviously, but we do kind of work with students and this is successful, so kind of along the same lines there. So, okay, so what does this all mean for you? Some of the expectations that we have. So we do have meetings pretty much every Friday, I want to say. When I first started, I was actually meetings every other week, but you know, we've gotten a lot done the past year. We want to keep doing that. There's still a ton of things to hash out. You know, we've never done next generation sequencing, but the closer we can get to that, the easier it can be for our next generation of recruits. So most Fridays, you know, there's a few things that might happen. The professors might not be available, but really check Canvas and we'll actually check your email more than Canvas because we don't check Canvas too much or updated it.
Don't you have a Discord too?
Yeah, we have a Discord. You know, I know a lot of you might be thinking, oh, this is a lot. This might not be right for me this semester. That's totally okay. Show up if you want, show up if you can. But yeah, so the Discord, I'll send that out in a few weeks once, you know, the club kind of downsizes and we have people who really want to stay connected with us. And then come ready to learn. So that's really important. When I first started out, meetings easily ran 5-6 hours and we were coming in when there was daylight and exiting when it was pitch black. And you know, that wasn't the most fun. And probably the professors didn't have the highest expectations, but now that we've gotten a lot done, I do have high expectations for you guys. So I want you to come prepared. I want you to read protocols that we send out beforehand, any articles. We do want you to have that knowledge before you walk into the classroom and start doing things that can potentially be dangerous. We're dealing with biohazardous materials, we're dealing with bacteria. We want you guys to be safe and know what you're doing. So please come beforehand prepared. And I didn't talk about this actually, but in our lab classes, so I took the biotech lab courses. They're a great way, highly recommend to take them if you're interested in research, you know, in a broader perspective. You're gonna learn more techniques than you do here. But in that class, we do have to handwrite our protocols before we can even come into the classroom. If you don't have those protocols handwritten, you can't do the lab. So, you know, this will be a little soft introduction. There's not too many protocols that we're going to be doing really. It's just going to be training, which will kind of be an offshoot. You'll learn some of the techniques that we usually do when we do DNA isolation with our water samples, but it's going to be a little bit of a different lab next week. So that's something that we want you guys to do. And I'll send out a little more instructions by email. And, you know, if that's a problem, if handwriting is a little hard, then, you know, please talk to us. We'll make adjustments. But really, the whole thing is about effort and being involved and, you know, coming in ready.
And reliably, if you say you're going to be here on time and be
Yes.
prepared. We have a problem with, because we're going to have to do some lab trainings to be able to even be involved in any of the actual project itself. I don't want to waste my lab resources and time training people who are just going to disappear or not show up when they say they are. So we are looking for some sort of commitment that, you know, you're going to stick it out at least through the end of the semester, right? And come when you can.
Yeah. And, you know, in the springtime, we get a few people who have they finished biology 400. They've, you know, if you maybe this seems like a lot, next semester can always be an opportunity to rejoin and kind of stay on the sidelines for a bit, see how everything is working out, and then kind of hop in later on. And really ask questions. So like Logan was saying, observing the water that we take samples from, and really asking, why is this happening? Why are we doing what we're doing? So in the field, that means noting observations like, oh, why is the water bubbling in this glass? It's because you're creating a high vacuum environment, and that's a lot of that pressure. You know, the water isn't warm to touch, but it will boil under that high pressure. And then in the classroom, when we do, I can show you guys the protocol. You guys will view it in a couple of weeks, but the protocol has a lot of steps. And, you know, sometimes it's like, why are we doing this? Well, you want to ask that question. You want to know the answer, because those little questions over time, that helps you understand the entire protocol and helps you understand the research in the bigger picture. So really important to ask, why are we doing what we're doing? Please ask. A lot of the answers are actually available to you if you search a little on our Canvas, in the protocols, etc. So yeah, that's our expectations. Are there any questions about that? I know a lot of people have classes on Fridays. Last year I actually had classes as well. I kind of worked with the teacher to go. He actually taught a different section, so I kind of asked, hey, sometimes can I come to this other section so that I can participate in this? And You know, a few adjustments had to be made just because this was really important to me. But otherwise, keep that in mind. Next semester will be the same Fridays, just because the professors are teaching classes Monday through Thursday, and then Friday is the only time that they don't teach. Yeah, any questions about that? Yes.
So you mentioned that when you first started, it was like 5 to 6 hours.
Yeah.
At work.
Okay.
Wouldn't be staying up that long.
Yeah. So in that case, we do want you to come, like if you're really interested in participating, you know, maybe in the future it might work out better that you can stay the whole time. We want you to come in, you know, maybe you'll work with somebody else and, you know, pass off your work to them once you have to leave. We do want to be respectful of your times. Of course, you guys are working, you're studying, you're balancing all these things. Super respectful of that. We understand. We just want you guys to, if you want to stay committed to this, please show up. Please let us know when you can't come. We take a lot of like some of the, when we send out emails and people tell me they can't come, that really helps me to see what type of materials we need to take out, how much of the aliquots we have to make. So that really helps out. But yeah, you know, if you have to leave a little early, we understand.
Just let us know. Communication is the key.
Yeah.
If we don't, don't surprise us. Let us know in advance, right? And it's not that every single, meeting is gonna potentially go all day Friday.
Okay.
It depends a lot on what we're doing, right? So when we were doing that was when we were prototyping a lot of the DNA isolation and the process where we had to go collect water in the morning, bring it back to the lab, fumble through the sample preparation and DNA isolation procedures, and then check that. And until we were pretty adept and experienced doing that, that takes a long time, right? So it's going to be variable. Like next week doing plasmid isolation, I don't expect it's going to take all day.
Yeah, no, luckily not anymore, especially if you guys come prepared, read the procedure beforehand. So I was telling somebody before this meeting that, sometimes we have to start at 8 a.m. Sometimes we start later, noon or 11, 10, whatever.
And some days we don't meet at all.
Yeah. And, that depends on what we're doing. If we're going out to sites, we're going out to water collection sites, then we're going to need to start earlier so that we can come back to lab and finish everything in a time efficient way. We actually do try to wrap things up by 2 on those types of days when we start really early. If we have like 10 a.m. start time, so in that case, like an earlier start time next week might help out with that. So we'll try to work out, and this might be a good time to ask, but what times next week would work for you guys? Like, does 10 a.m. sound good? Yeah. Great. So yeah, in that case, you'll be able to stay for a lot of it. We do have to lecture a little bit beforehand. I'm still thinking if maybe me and Logan this weekend can work on like creating like a YouTube lecture that you can watch beforehand, just so that you come in with everything that might make our time shorter explaining the lab to you. But we'll see about that if we have the time.
Yeah, that's why preparation is really important, and like
Yes.
reading the things ahead of time, and so you can come in and immediately ask any questions you have or things you're confused about. Because otherwise, if we come in and nobody's even looked at the procedure yet, it's going to take us an hour just to explain what we're doing and kind of read through the procedure before we can even get started. So that's what leads to those super long lab days.
Otherwise, for our old group, the past semester, it wasn't taking us very long. We could actually finish our DNA isolation once we had the filters. We could finish that within like an hour and a half. And, it's going to take us a little longer starting off. We have so many people, so many hands. You guys are going to work in groups, but yeah, it really depends on you guys how long that takes. Yeah. And then, so necessary materials. This semester we have to be a little more careful about our lab safety. So if you're in a lab class, you probably already have a lab coat. We don't want this to trouble anybody. Honestly, a really large t-shirt, like a button up or
Well, not
something.
t-shirt, it's got to cover your arms and
Yeah.
go down past your waist, and it has to button or zip in the front. So there are some rules about that. I can look, I might have a few lab coats, but in the cabinet, but they're old and dusty, and I definitely don't have enough for everybody, nor can I guarantee sizes.
Yeah,
So
but
if you plan to go on in the lab, it's good to have a lab coat. You'll probably need it for other courses because we require that, like in our microbiology classes and in biotech labs as well.
But I think even like in past classes, like for anatomy, when I took it, they said if you have like a button-up shirt that's really long on you, of course you don't want it to get in your way when you're doing lab stuff, but that works too for a little bit.
Yeah, and to just talk to us about that if you think you want to go that route in lieu of a lab coat, because there's just some things that have to be true about the shirt, but that is an acceptable way to cover that.
Yeah. And then safety goggles or glasses. You know, if you need a pair, we sell some in the chemistry stock room, but otherwise you will need some sort of eye protection wear.
And it needs to cover the sides, right? It can't just be in the front. It has to wrap around. Ideally, it's like the chemistry goggles that you have from your chemistry classes, right? So if you have those from chem, those will work. And technically, it's supposed to be the splash goggles.
So again, if you don't have that, Amazon has cheap alternatives as well. Shouldn't be too much. Gloves as well. We can't really provide, you know, everybody's different size gloves. Just go the regular nitrile. If you need like a picture, like these types of gloves, you know, talk to us after class if you need to figure out what type of gloves are right.
And
But-
don't get powdered.
Don't get powdered, and try not to get latex because, you know, some people might be allergic. We don't want any allergic.
You can use
reactions.
latex if you.
I'm allergic to latex, so please
Okay.
don't. I won't be able to help you too much with my bare hands otherwise. And then this is kind of a soft requirement. It's really helpful when you're working with some of these tubes.
Lab attire.
Oh, yes. Yes, lab attire. Hopefully you guys know this from your other classes, but you know, long pants, closed toe shoes, shirts that cover your shoulders. Anything else?
No, it's mainly the closed-toed and closed-heeled shoes, and the top of your foot also can't be showing. So I know some people wear shoes where the entire top of their foot is sticking out, but technically it covers the toe and the heel. That's also not acceptable. Basically, we don't want to see any skin at all from your waist down.
Again, you're also wearing the lab coat, so you know. But I know it gets hot sometimes, but you know, this is a lab. We do want you guys to be safe.
Working in the lab is like you have to wear a uniform, and this will be true in any future lab thing you do. When you go to four-year institutions, when you're working in industry and healthcare environments, right? The appropriate attire is really critical, especially these days. They clamp down on it a lot, so be ready for that. It won't just be here.
And then soft requirement, when you're writing on tubes and stuff, labeling them with what the sample is, really helpful to have a fine point Sharpie. You won't be able to read if you use like a regular big Sharpie, and it can be a hassle sometimes to be like, hey, can I borrow this Sharpie real quick? Like other people are going to be using it as well. So if you can find one around your house, a fine point Sharpie would be really helpful. And then technology to view protocols. So obviously, like I said, we do want you to handwrite your protocols, but it's not going to be perfect. You're going to get yourself confused at times if you do that. So please bring some sort of technology, iPad, computer. The library, I actually use a Chromebook as well from the library. It's right over there. So if you don't have something you can bring, like go over to the library and check it out for the semester, a regular Chromebook, and that'll be fine.
You don't want something too big though, so keep in mind we're going to be doing lab stuff. So you don't want to have your computer out and then you're going to spill reagents on it. Right?
Yeah.
Because it's right there in the lab. So that's one reason to have handwritten protocols. Although we do keep our lab notebook electronically, so you will want to have some sort of device where you can, access the lab notebook, for recording what we were doing and adding details.
Yeah. And then for the next lab session, we actually want to get right into it. We're going to do a plasmid isolation lab. So as you know, we isolate DNA from the water samples, but in this case, we're actually just going to take a bacterial sample and isolate the plasmid DNA. So that's a circular piece of DNA within the E. coli cells. And that actually has a green fluorescent protein gene inside of it. So we're going to isolate the plasmid and extract the GFP. Actually, no, we're not extracting the GFP. No, we are. Yes, we are.
We are.
Through
We're
columns.
going to do HIC.
Yeah. No, sorry, no. Just we're gonna purify it.
Just the
and-
DNA, not
yeah.
the GFP. Just
Yeah.
the DNA molecule that has the coding sequences.
Yeah, I forgot we're not doing restrict and digest. We'll start working on the lab stuff. It's gonna be biohazardous materials, so come prepared with some of the materials. If you have any questions about that, please ask. But otherwise, I'll send details about that in another email. And yeah, is there anything else? No? Yes.
Sorry. Are you sending it to the email that we put in for the QR code?
Yes, and we'll also add you guys to the Canvas. It's not the cleanest, so I'll direct you guys if you need to see last year's introductory recording. I'll point you guys where you can find that. Maybe it's kind of hard to find, but
There's a lot of other cool stuff there
There's
too. We've
tons.
had lots of guest speakers that we've recorded like during the pandemic when we couldn't meet in the lab. So you can go back and watch all of those speakers.
and you can access our lab notebook. I will say about the lab notebook, it's right now it's accessible for everyone to use, but please Be respectful, don't change it around too much. If you accidentally do something on it, please click undo. And just so we keep it neat. Yeah. Anything else? Yes.
If you can't come to a meeting, you want us to email you or
Yeah.
tell you the week prior?
I mean, we understand things happen on the spot. Sometimes we can't make it. As early as possible, just because we do aliquot some of these solutions in here. So, we don't want to over aliquot and then, these solutions go to waste. So knowing how,
Best.
yes. I mean.
Yeah, if
just
you
the-
tell me, I may or may not remember to tell you.
Yeah.
Okay. So
Thank
do you
you.
have an email? Let's see.
Yeah, so on the flyer, it has my email. I'll also send out an email to everybody with their, their personal or W emails on there.
They should be able to just reply to that. one.
Exactly. And save me as a contact or whatever. Yeah. Anything else? Yes. Good.
No, so previously we haven't had to require this sort of stuff because a lot of these kind of techniques, historically, not everybody does wear all the PPE, but because we're starting to grow and we are working with some potentially hazardous things. And in our lab, those who've taken lab courses, you might notice this semester we're suddenly requiring long pants like we didn't used to. So that's kind of filtering down to the clubs and things also that are doing anything in a wet lab sort of setting. So I'm not going to be super picky about it. I feel like as long as we appear that we are trying to follow the rules and do it properly, right? So as long as everybody's got, shoes that enclose their feet completely, their legs covered in some way, shape, or form, so there's no skin showing, and have something that is either a lab coat or a facsimile of a lab coat, as well as the eye protection. I think that's fine.
I was gonna say Spirit Halloween. Like sometimes I've used that in the past for quick lab coats when you needed it. So
Yeah, I think like these days you can buy it in lab coat on Amazon for like $12.
Yeah.
there's a so
And bonus with that, you can use it as mad scientist.
I've actually
There you
worn
go.
my lab coat as a Halloween costume.
exactly.
Yeah. Anything else? You can always email me. Yeah.
Can you talk more about the Sac State grant thing you were talking about earlier?
Yeah. That actually might be best.
Oh, the serious. Well,
Yeah.
that's actually just a long-term project that has been going on at Sac State. It's NSF funded, the National Science Foundation. It's to try to get research experiences into classroom. So it's actually the Serious 2 grant. The first one was actually back when I was a lecturer at Sac State, and in a number of their different core biology courses, they had a sort of similar water project where they were just, instead of doing metagenomic sequencing, they were just cloning individual 16S ribosomal DNA sequences and sequencing them. But they actually had it set up where it would go through several classes where like the introductory bio students would isolate the DNA, then the genetics class would PCR it out, and then clone it, and then they would pass it off to one of the upper division classes for sequencing. And that was pretty successful. So they wrote another grant and got the Los Rios colleges involved. So we're trying to, put those kind of research experiences in various other courses. So I actually have a project that we do in one of my biotech lab courses where we do bioprospecting. That's part of the serious project, where students bring in soil samples. Then we dilute it in a buffer, make serial dilutions, plate it out looking for colonies. Then we start to characterize the microbial colonies from different types of media. And then we screen them for enzyme activity. And if they pass our qualitative screen, so they have a useful enzyme, we focus on amylase, which breaks down starch. Then we take those strains and then we try to quantitatively do more sophisticated enzyme assays to kind of measure their enzyme activity, and then compare them to known amylase producing strains, right? And this is something that you see in the biotech industry, because there's companies that go out and, they're screening soil microbes looking for new enzyme activities or things like that. So that's kind of an example of the sorts of serious projects, although they span all sorts of disciplines, not just biology. Those are just ones that I'm familiar with.
Yeah.
And we're not exactly directly related to that. We're mostly related to Sirius because they have a student research conference, which is where we presented the posters. And I'm involved because I do some in my courses also, because the grant itself is mainly for in-class things. Although I might take some of the stuff we're doing here and eventually incorporate it into my courses. But I'm hoping that grant will actually buy us a nanopore DNA sequencer this year. We'll see what that goes.
So you don't use any of the facilities at Sac State?
No, not directly. I mean, some of the people involved in the project do, but we don't. We mostly do all that stuff here.
And then, you said it's through most rows, like all the colleges, is ARC the most like active within the?
Yeah, but that's just because we're the biggest. So there's more faculty here that are involved in it. But like there are folks involved in it from, at FLC, Sac City, CRC.
Well, thank you.
Yeah. And just a real quick offshoot, classes like Chem 309, that's for nursing students, Chem 400, they're actually involved with that series project too, and they present posters alongside of us. So there are classes here that are also involved with getting research into the classroom experience.
Yeah, so actually, if you've had one of those lab courses recently where you did some little project like that, that might very well have been part of the serious project.
I'm willing to check this.
Okay.
But I would recommend that we do Fridays.
Fridays are just the only day that we can do it because there's classes in here Monday through Thursday, and I'm pretty much booked with all of my teaching schedule Monday through Thursday. So Fridays are really the only time that most people have free. So we realize it doesn't work for everybody, but just logistically speaking, Fridays are basically the only time it works. So that's sort of what we're stuck with. So if you want to participate this semester, then you might need to change your work schedule if you're normally not available on Fridays. And we realize we can't accommodate everybody, but it is what it is. We're just trying to do the best we can given the situation.
Then I know if some of you guys are in organic chemistry, they usually do like a support class from 12 to 2, I actually did that when I took OChem, and I would just grab the worksheets. I'd, check in at 12 p.m. I'd grab a worksheet and, do my own thing. If I had questions, and the next week I'd go in and do my thing. So any of you guys are in that, just consider, other places can be flexible as well.
Yeah, and just communicate with us too, right? Because like we have had plenty of meetings like in like last semester where, folks had something going on in the later afternoon or had to go to a class or something. So they come and, do as much as they can with us, but they let us know in advance that they're going to have to leave at a certain time. Or, some people, oh, I can't be there right at 10 when you're starting, but I can be there at noon. Well, to pick up where you are there. So the key thing is to communicate with us in advance about what's going on. So we're not like, wait, where are you? You said you were going to be here. Right. Or, things like that.
And, we'll do recruitment again next year. I'm going to be transferring off. Logan will be transferring off. So we need more people to be settled into the club this semester. But again, there's always going to be recruitment in the fall. We get a few people in spring. We don't advertise it in spring. It's really just for people who have done who are starting out with the biotech courses, or they're, completed a class and they really want to jump in. So spring can be a better option for some. Yes.
I was just curious, the research conference you're talking about, what, kind of conference is that? Like, is it through like community colleges or CSUs or national?
Well, that's a serious conference. So that's through that serious NSF grant. So that's primarily Sacramento State and the Los Rios colleges at that one. So it's mostly like the sort of local stuff here. Right. We definitely potentially could go try to, present at other research conferences, but we wanted to develop the project a little more before we, kind of took it out of the more local area. So once we start getting metagenomic sequence data, right, I think that's when we're going to start to consider, trying to go to, other research conferences outside of the local stuff.
Yeah, actually we applied for a research conference at Stanford, but we weren't accepted at the time. Hopefully as we continue our methods, we can of course apply anywhere. There's so many throughout California, and we're open to doing them. We just have to, continue our research a little more, get you guys involved. So that's more of a springtime thing.
Got to make sure we got something good to present if
Yeah.
we're going to do it.
Yes.
It's not a process about.
So basically, you say who's going to be attending, you want to talk about your research. So what we've done so far. So when we did that Stanford research application, we had to give what was our research about. We had to actually give our CVs as well, curriculum vitae. So they want to see what you're up to. So that's kind of the standard thing.
Yeah, you always submit an abstract that's a
Yeah.
description of what you're going to present. You don't have to share your actual poster or whatever, because a lot of times people are working on that right at the last minute beforehand. But you do have to tell them approximately what you're planning to present.
Yeah.
Right? And it depends a little bit on what the conference is. It's a little bit variable and isn't quite the same from conference to conference.
Yes.
For research projects, a lot of the ones you guys are doing are the microbes in the water. Would you ever like consider doing the research like maybe on the plants and the vegetation around the water or the soil? I know you might have already done this.
Well, we're considering that for sure. I mean, my main interest was kind of developing the pipeline for the water specifically, right? But that's the kind of, interdisciplinary and other like collaboration stuff we're looking for, other folks who are interested in leading the charge on those kinds of things. We're hoping that with the microbial sequencing from the water, we'd like to actually expand that to look at like eDNA or environmental DNA. So we can, look at, traces of fish populations that are in there or other invertebrates in the water, right? Or expand it to looking at the soil on the shore banks and, and combine our data with the organismal biologists and ecologists, like tracking things going on the sides of the river and in there. So we'd love to do that, but we're not there yet. But if folks are interested in that, that's definitely something we can talk about pursuing.
Awesome.
And then a question we've gotten in the past is whether this can count as class credit. Now you have to talk to your other professors about that. This is not for credit. This is really your own experience, something to add to your resume, something to get behind you. So this is not like course credit at the moment. So if you want to use it as something outside, that's on your own professor's accord. So.
Yeah, and we sort of call ourselves a club, but we're not.
Yeah.
We at least not for a while have we actually been officially a club.
Yeah. We're not an official campus club, but this is a research project. I call it club as a formality for students, and it's accessible to others. So.
Is that a good thing that it's not officially a club? Because like we get more like
There's trade-offs. There's
Yeah.
reasons to be a club because then we could get a modest amount of funding that might like say buy us all pizza at a meeting sometime. Not that we can blab or anything, but things like that. But that comes with some strings. Everybody, at least one representative from the club has to attend these regular meetings of all the other club officials. So there's some extra bureaucratic overhead, right? But the carrot that they're dangling for doing that is like a modest amount of funding. But since we mostly are in the lab and can't really utilize that, the funding for food and drink at events and stuff anyway, we've kind of just said, we don't want to make anybody waste time going to those meetings. Let's just do it unofficially.
So when it comes to funding, what is this club classified under?
It's not. We're just a research project. We mostly run from various modest small grants and just basically lab supplies and materials that I donate from our biotech program. Right. That's one of the reasons I mentioned commitment earlier. We're going to start out burning through a bunch of expensive DNA isolation kits and using expensive reagents. I don't want to use all that to train you just to have you disappear and not be around when we actually get to isolating the water samples that we were presumably training you to help us with. So not that I don't want to scare people off with that, right? But we hope that people don't just sort of only casually engage with this, where you just show up when you feel like it. And right, we want people who are going to come down and get in the trenches with us and help us actually push this project forward.
I had one other thing to say, but I've forgotten it.
What's this?
Oh, this is, if you're here at the beginning, you probably already filled this out. This is just a little survey so that we can get your name, your email, your W number, add you to the Canvas. Please do this if you haven't already. That's how we'll communicate with you. Again, you can sign up now. If after this information session you think, oh, this might not be for me, feel free to, still fill this out, stay updated for a little bit. We do tend to downsize the email list by the end of the semester just so that it's clean and we know people who are actually involved. So, over the course of the semester, you might stop getting emails if you stop showing up. So
You probably will stop getting emails if
Yeah.
you stop showing up.
okay, so we will stay behind. For people who don't have lab experience, we actually want you to get started on micropipette training just so that you're ready for next week. If you haven't taken biology, chemistry, that's totally fine. We'll take a quick break. You can ask questions.
Actually, can I make a few comments
Oh,
as
yes,
you're wrapping
absolutely.
up before you go into that about the stuff afterwards?
Yeah.
So we're going to be using a lot of biotech tools, including micropipettes. So some classes use the micropipette. So what we were thinking about doing today, because we haven't done all the lab safety training, because there's going to be a bunch of paperwork that we're going to have to do and spend a chunk of time before we can start doing lab stuff next week. basically going through the lab safety procedures and, you have to fill out the liability waiver similar to the lab courses and all that stuff to participate. So we're gonna start out doing that. So if you don't show up on time next week and you miss the safety training, you can't show up late and participate because you haven't gone through the lab safety training, right? And the ones for your other classes don't count because that's specific to this lab. So just be aware of that. Yeah. We had floated 10, but I don't think that's firmly decided yet. We were going to check in with the group and try to figure that out. So that's kind of, I don't know if we were going to try to figure that out today or if that was going to be in an email about the meeting next week based on the responses to the survey.
Yeah, so Logan here, he has something that like 2, right?
Yeah.
2, so, and you're second 2, so, we want to start as early as possible. I don't think I can wake up before 9 AM, honestly.
Oh, my wife, I can't reliably be here to start unless we're going to a site to collect before 10. So
Yeah.
10 is usually our sort of default start time. We figured this time 11 was a little better because we, might get more engagement for the first time.
So 10 work? Okay, great. I'll be our start time next week.
Oh, and so for folks who have used micropipettes, we're going to use them a lot. So we thought today we can just do some refresher training for anybody who wants it or anybody who's never used a micropipette before. You've never heard of a micropipette, that probably means you should stick around for that. Because that will be kind of required to going forward. However, if you already know how to use a micropipette, you're welcome to stick around anyway and just get some additional practice with it. But that's all we're going to kind of do afterwards, just kind of hang around and chat for a little bit and get some practice with the micropipettes. And then you'll send out the procedure for next time. But we're not going to start that right away. Again, we're going to begin next time with some lab safety training and paperwork. And you will want to make sure you respond to this so that not only do you get information about next time, but you get added to our Canvas, because we're probably going to have to do electronic versions of the safety paperwork, because the administrators usually want that in Canvas. And that'll be in addition to the hard copies that we'll do in class. So you do need to make sure you get into our Canvas site so you'll be able to do that and participate.
So otherwise we'll take like 10-minute break, 15. Yeah. Okay. Yeah, however long when you guys are ready. If you need to leave, please do whatever.
Were you going to do introductions with everybody? Have folks introduce themselves? Because I know we talked about kind of doing that as we wrapped up before we took a break and got the pipettes out.
Yeah, so I want to put a name to your faces. If we could actually, if you guys have the time, if you have to leave, please do. Otherwise, introductions. Oh, if anybody came in late, I'm Anuya Banerjee. My name is spelled out over there, and I'm the Real STEM president. Right time, right place, right motivation. I love saying that. So I've been here participating in Real STEM for about 3 years now. I've been at ARC for 7 to 8 years, taking classes here. This is my second home. Love it so much. I'm also, if some of you guys are interested in like campus, like life, per se, there's a lot of great clubs here, actual official school clubs. So there's the Take a Hike Club. I'm actually vice president for that. There's AMSA if you're on the pre-med pathway. I'm treasurer for that club. There's also Women in STEM if you guys are interested in a group of people who, all women. There's probably a few males in the club, I don't know. But if you're interested in that, they do field trips, they do activities, they do a lot of fun stuff to get people excited about science. So that's over in the STEM home base area. So honestly, if there's any resources, any questions you have about campus, I can definitely answer. If you need like food services, the food pantry, they do actually like a quick produce grab a couple times a month. I just got an email about the dates. So, you need anything on campus, I will be your person and help you out. And then that's Logan.
I'm Logan. Yeah, I'm third year at ARC, 1 year into Real STEM. Again, same right place, right time, right motivation. Great saying. And yeah, for this club, you really do have to be motivated. You really want to do this research, want to progress it. So yeah, we're looking for those people.
Yeah, I encourage you guys to find your own motivation for why you're participating in this club. It might just be, okay, I want to get some research experience behind me, have that leg up versus other students. And, that's great. My motivation, I kind of figured this out as we go along. I used to be a diehard, or very hardcore germaphobe, and kind of studying the water has helped me, understand like there's really only hundreds of pathogenic bacteria. They're not all bad. We're obviously collecting water. I've actually fell into the water at one of these sites, and like, or past me would have been terrified at that fact. But knowing what I know about what's in the water, so far we haven't found anything crazy. So
I don't know. I'm pretty sure there are some hazardous microbes in the water. I'm pretty sure there's some of those serious projects that actually isolated some antibiotic resistant bacteria from the American River.
okay, it's not all great, but so far, that I might revert back to my old ways. But for now, we're okay. Yeah, so find your own motivation for why you want to participate.
And I'll mention really quick, like, I'm not someone who has taken the biotech courses. I really want to, but I haven't gotten around to doing it. I came here after my Gen Bio class, really excited, really passionate. There's a lot you can learn from doing this kind of research and just participating. There's a lot you can learn from conferences, from doing practice, from your peers, the professors. So I encourage you, like, if you're coming here, like, come here to learn. It's what we're trying to do. So yeah.
Yeah, I think what we mean, just to build on that, what
Yeah.
we mean by like commitment is that, you've got a desire to be here and you're going to actively like engage and really like, participate in it. It's not something that you just kind of casually show up for.
Yeah.
Right. Because you may, need to do some stuff outside of the times of our meetings, right? To help us out. And a lot of times we need to research something. So oftentimes we send people off to do that.
And some people, like one of our past members, he's still on campus. Oh, real quick, we do have members for last year as well. So there's about 6 of us who are continuing on. In the background, they might be working on some stuff so that we can streamline our process for you guys this semester. Hopefully they'll join us for future meetings. Right now, only past members would be me and Logan, but there are a few others, so they'll also be helping you around lab and stuff. So yeah. And then-
Do you want to give a break or do we want to do the
Yeah. Introductions actually. I would love to hear your name, your major, what you're studying. Maybe if you've had some science experience, you probably already said that here, but name to face, that really helps me. Fun fact if you want to.